Welcome to our first science seminar for the semester. And we're going to start out with something exciting here. Graphene and quantum dots. Tatum Martin is going to be talking to us about these things. She's one of you, a student. She's going to tell us about the exciting... Nice to see you again. No, we got to talk. Okay, don't run away. Yeah? Don't let her run away. Okay. So, what was that?
Exciting seminars.
Exciting seminars, yes. We have an exciting seminar today, and all through our semester here. So, Annabelle Malinowski will be presenting next week on some biology work that she did as an undergrad. So we'll have physics today, biology next week, and lots of other exciting things through the semester. If you want to know what's up for the science seminar, go to letu.edu, and then the / the one that leads forward, yes, and then type in science seminars, and you'll come up with this scheduled seminar for this semester. All right. So today, Tatum Martin, she's a transfer student from Texas State, which was where again?
San Marcos.
San Marcos, yes. And so she's happy to be here and not there, apparently, otherwise she wouldn't be here. And she wants to be with you guys because, you know, she heard so much about wonderful people at Laterno. And so welcome her to her time here.
To do a research experience for undergraduates at Texas Christian University, and I was working with Dr. Anton Nomov. We were studying graphene quantum dots, photothermal cancer therapy with molybdenum disulfide and thulium. To give you a general idea of what we were looking for was we wanted to find a dopant for our specific graphene quantum dots that would absorb in the near-infrared region. So a dopant is something that we're adding onto or attaching to our nanomaterial, which is our graphene quantum dots. And we were looking for the near-infrared region, and I'll get into why that's important in just a minute. After we were going to find that nanomaterial that was doped, we wanted to evaluate it based on how much its temperature would change over small amounts of time. And then we wanted to look at in vitro experiments and in vivo experiments, which would be in vitro is in cell experiments, and in vivo is in animal experiments. When I typically start telling someone that I'm working with nanotechnology this summer, I get a lot of, oh, you're making those robots that crawl up your arm and go kill the cancer cells. That's not exactly what we were doing. Well, it's a great idea. But we were working on carbon-based nanomaterials, which is what the graphene quantum dots are. They're 3 to 5 nanometers long, and they're very much like a sheet, and they have functional groups on them, which makes it easier for us to add those dopants onto them. Some of their properties are that they have a high quantum yield, that they have low cytotoxicity, and they have good biocompatibility. And the reasons why those are important is because of what we're using them in. So biological imaging and sensing is utilizing the it's utilizing the property of how it emits a different wavelength when it is irradiated with one. And the fluorescence, I'm sorry. So fluorescence is when it's irradiated with something else and then it emits another wavelength. And what's good about that is we can irradiate the skin or the cells with a certain wavelength, and then we can use microscopes to see the other wavelengths that the quantum dots emit, so it shows us where the cancer cells are in the body. And the reason the cytotoxicity and the biocompatibility is good is because it is going into those cells, so we don't want it to harm the cells. We want to be able to use it without hurting that. And to explain the quantum yield, imagine if you threw a ball down a long tube. Every time it bounces, it's losing some energy. So when we're irradiating the quantum dots, we want them to emit the same amount of photons that were given to them, and that gives us a high quantum yield. So not losing energy as they go through the material. What we use the quantum dots for, though, is photothermal cancer therapy. And to give you a better idea of that, what's going on is we're putting the nanomaterials into these tumors, and then we're irradiating them with a certain wavelength. And what happens is the nanomaterial absorbs it, it heats up, and it starts to kill the cancer cells that way. So the reason why we want to use near-infrared wavelengths specifically is near-infrared is from 800 nanometers to 1100 nanometers. And what's good about them is that at low power densities, they're not going to harm your cells, but what they can do is they'll penetrate much deeper than other wavelengths. So in the future, they're looking at using this with brain cancer treatment because it's less invasive. So instead of having to surgically remove the tumor and go all the way into the brain, you could irradiate and it would pass through that tissue and reach those nanomaterials that way. The other thing is these nanomaterials aren't going to harm your cells the same way cancer does. Cancer is killing all of your cells, hopefully your cancer cells faster, or chemotherapy is killing all of your cells, hopefully your cancer cells faster. But the photothermal therapy is supposed to not do as much damage to your healthy cells. What we found was we wanted to look at thulium NGQDs, molybdenum disulfide NGQDs, and GQDs. So NGQDs are nitrogen-doped graphene quantum dots, and GQDs are graphene quantum dots. The difference is in how they're made, and I'll get into that in just a minute. Before we could start making them, we had to figure out what materials had those near-infrared absorption that we're looking for so we could dope it onto the graphene quantum dots. So we had to look at their absorption spectra. And absorption spectra shows how much of the light is absorbed versus what wavelength it is. We used two different machines to do this. One was the UV Vis, and one was the Avantis. And the UV Vis excites from 200 nanometers to 1100 nanometers, while the Avantis excites from 400 nanometers to 1100 nanometers. And what's good about the Avantis is even though it has a smaller range, it gives us much more clear depictions of what's going on, so we can see more peaks there, but the UV Vis gives us a wider range. We looked at three different kind of categories from literature of things for near-infrared absorption. So one was nanoparticles, one were rare earth metals, and the other was some various dopants. Out of the nanoparticles, cerium oxide, copper sulfide, and silver sulfide were what we tried. And none of those actually showed any absorption in the near-infrared for our samples. But with the rare earth metals, we did see good near-infrared absorption in erbium, ytterbium, and thulium. Praseodymium, samarium, and holmium had good absorption peaks kind of further in the visible spectrum, but not quite into the near-infrared, which is where we're looking. And then the other dopants we looked at were molybdenum disulfide and tungsten carbide. Both of these are hard to dissolve in water, and it's important that they're dissolved in water because of how we make them. So I'll explain more of that in a minute, but tungsten carbide we could not get to dissolve at all, and so we couldn't use that even though it has good absorption in the near-infrared. This here is a graph of the absorption spectra for erbium and GQDs. So you'll see that you have the absorbance in an arbitrary unit on the y-axis and the wavelength here in nanometers. Remembering that the near-infrared is between 800 and 1100 nanometers, this is kind of the area that we're looking in. We see two peaks that I noticed were important to me was a peak here around 800 and a peak at 975. The reasons why those two stuck out is because the two lasers that we have to use in our lab are an 808 nanometer laser and a 980 nanometer laser. So while they're not exactly at either of those lengths, there might be enough of a peak there that we could see some absorption at those lasers that we have. For our ytterbium NGQDs, we see a peak over here at 975. Again, just looking in that 800 to 1100 range. For thulium NGQDs, here's where our 800 starts, and we go into the 1100. So we don't actually see a peak in the actual near-infrared region. What we do see here is a peak in the shoulder at 790 nanometers, and that we thought might be close enough to see some photothermal properties in the thulium NGQDs. And then for molybdenum disulfide, we actually don't see a strong peak at all in the near-infrared region. But this right here is 0. So we're seeing good absorption all the way through that near-infrared region. So even though it's not a peak at where our laser is, we thought there might be enough absorption there to see some denaturing of our cells. And for the GQDs, the reason why this graph looks different is it was on a different machine. And so in the 800 region to 1100, again, not a strong peak, but there's a broad absorption. So we thought we might give it a try. When we make the GQDs, we start with either glucosamine or graphene oxide. So if you'll remember, I mentioned that there are nitrogen-doped graphene quantum dots and graphene quantum dots, and their difference is what they're made of. Nitrogen-doped graphene quantum dots, their precursor is glucosamine, and graphene quantum dots have a precursor that is graphene oxide. We dissolve that into water and add whatever our various dopants are, and then we microwave it for about an hour. After it gets microwaved, we purify the sample with a dialysis bag. And what's happening in the purification process is any glucosamine or any graphene oxide that did not become a graphene quantum dot is getting filtered out of the sample. And so you can see a clear difference here in the color. A much darker sample was not purified, and then after purification, it's a much lighter sample. We needed to determine the concentration of these samples as well. So we used 6 cover slips per sample, and we would mass those here on this machine. And after we masked them, we would put 40 microliters of our graphene quantum dot sample onto each of the cover slips, and then we put it in an oven for 24 hours. After 24 hours, the water had evaporated from the sample, and we were left with just those quantum dots, and we were able to mass them again and determine their concentration mathematically. We wanted to look at temperature change. That's the big thing for us in photothermal experiments, is we need those nanomaterials to absorb that light and to heat up and change temperatures in order to kill the cells. So we decided we were going to experiment just on their photothermal properties, which is really just how much they change temperature. Our goal was looking at a 5 degrees Celsius change. So the human body is 37 degrees Celsius, and proteins start to denature at about 42 degrees Celsius, which is why we're looking for that 5 degrees. What we did was we put 1 milliliter of sample into a cuvette, and we used a thermistor, which is what this here is. It's insulated, and it goes into the cuvette, and we capped it, and the thermocoupler is what gives us the temperature here. We chose 1 milliliter of sample in that cuvette because the area of the laser that it was lighting up is about the same as 1 milliliter of sample was in our cuvette, and we wanted to make sure that we were heating it thoroughly and all in the same area instead of heating it a little bit at the bottom and then it having to move to the top eventually. We chose to use water as our control for what we were comparing with because water is the base of what all these nanomaterials are in. And so we wanted to know, is it the nanomaterial that is absorbing and heating up, or is it just the water that's absorbing and heating up? So we needed to compare that as well. And we did this experiment for 20 minutes on each sample. Then we had to evaluate it. So we look at these two graphs of the water and its change in temperature. Again, remember that we're looking for a 5 degrees Celsius change. So we see a 0.7 degrees Celsius change from the water at 808 nanometers. That's not too much of 5, and compared to the other data we had from our samples, it was not overtaking in such a way that we couldn't tell the difference between the nanomaterial heating up or the water heating up. However, when we looked at the water at 980, we see a 2.2 degrees Celsius change, and that is much more of 5. And so we decided that we didn't want to keep looking at the 980 nanometer because we would be just heating up the water, and it would be harder to tell, is this the nanomaterial that is causing this, or is it just the water's photothermal properties? Then we started looking at our samples. So for our thulium NGQDs, our molybdenum disulfide NGQDs, and our GQDs, we decided to choose these to work with because the thulium, we see a 2.7 degrees Celsius change. It's not quite the 5 that we're looking for, but we thought it might be enough that we would see some denaturing of the proteins in those cells and that we might be able to cause some damage to the cells. For molybdenum disulfide and the GQDs, we see a 3.7 degrees Celsius change. Again, it's not the 5 that we're looking for, but we thought it might be enough, and so we wanted to give it a try. Before we could go put these samples into cells, we needed to figure out how toxic they were to the cells. And to do that, we did an MTT assay. So MTT is dimethylthiazole. And what happens when you put it into the cells is it goes through a process, and one of those products is called formazine. Formazine needs to be dissolved with DMSO, and when it is, we see that it has an absorption peak at 590 nanometers. So what we can do is we can run these through a spectrometer, and we can see how much of that light is being absorbed to know if there is a lot of the cells are alive, or if not very many of the cells are alive. Here you see the results from it. These are our graphs. You see the cell viability in percent on the y-axis and the concentration on the x-axis. What's important to know here is that when these cells come out of the incubator and you give them the MTT and then you put the DMSO in there and then you run them through the experiment, they're out of their happy place for a long time, and so they're going to die a little bit. So we had one sample that was a control for each of these tests, and that one sample was only had DMEM in it, and DMEM is the medium that is what the cells live in. And so however many of the cells were alive in that was counted as our 100%, and so everything else is in comparison to that one sample. What we're looking for here is to see 80% viable cells or more. So their cell viability is how much of them are alive. And we're looking for 80% because that's the standard in the research that we're doing, is you choose 80% viable. So here we had to do 6 serial dilutions on each of these 2 samples, the graphene quantum dots and the thulium NGQDs. The molybdenum disulfide, I mentioned earlier that it was hard to dissolve in water. We had to add a surfactant to it, which makes it much more toxic to the cells in order to break down that molybdenum disulfide. So we actually had to do another set of serial dilutions on the molybdenum disulfide to see any. So it's much more, or it has a much smaller concentration than the other 2 samples that we had. And serial dilutions are also done with DMEM, which is that medium again. So that brings me to this next point, is a lot of what the cell is in is DMEM instead of water. So when we did our photothermal property experiment earlier, we were looking at water as our control, and we didn't think of DMEM. So we had to come back and say, okay, how does DMEM change when it's irradiated with that laser? So we chose to test that. And what we actually see is a decrease of about 0.5 degrees Celsius. And I think the reason that we're seeing that is because the DMEM has to be warm to go into the cells. So it's sitting in the incubator. It's a lot warmer than the surrounding space. And the experiment that the photothermal properties were running is a cold metal box that's blocking off any light and heat for safety reasons, and also to keep it more accurate. But we go from a very warm temperature to a very cool temperature, and so I think what you're actually seeing in this region right here is it just adjusting to the temperature of the box. And then after that, we don't see any real change, about 0.2 degrees Celsius. It varies in the rest of the time that it was in there. Now we get to look at going into our cells. So what we have is our control, which is the DMEM. It's just the medium that the cells are in. So it's, if you had the cells and you weren't experimenting anything out of them at all, that would be what was in the control. And then we have one where we're adding water to it, because again, the solution that the graphene quantum dots are in is mostly water. So we wanted to see how water compares. What we have here is the lowest concentration we use of our sample. That low concentration is the same concentration that gives us 80% viable cells. The high concentration is what we used in the photothermal properties experiment. So when we were originally figuring out the temperature change, that's the high concentration. And then from there, we have that 80% viable cells. We also chose to irradiate the top row with the laser, and the bottom row we did not. So we could compare between how a laser irradiated cell and the one that didn't get the laser irradiation would show. And this is done in a 96 well plate. The 96 wells were chosen because the area of the wells is about the same as the laser area. So again, we're looking for that even heating of all of the cells instead of a where it's got to heat up in one area and that thermal energy has to spread. This is just part of a well plate. There actually are 96 physical wells on a 96 well plate, but I'm just showing you what we did. And we spread them out between two, so that way when one was heated up, the other cell wasn't in its surroundings. It wasn't going to be affected by the temperature change there. Now that 96 well plate has to have a lid on it because we don't want to contaminate the cells that are in it when we're putting it into an oven later, and then back into the incubator, and then to the MTT assay. So there's a lot of going through, and for antiseptic purposes, we needed to have a lid on there. That lid's going to be between the cell and the laser, so that brought to mind a question of, okay, does the lid absorb at 808 nanometers? Is that going to affect our data at all? So we ran it through the UV vis, and we see here at around 808, and it's very, very low absorption, and so we were not worried about the lid interfering with that experiment. So we get into this experiment now. And this experiment has to be run as quickly as possible because we're irradiating 4 wells per plate that we use. And so it's 20 minutes per well. And then there's also time in between when you're filling with the sample, when you're putting it in, when you're cleaning it off, and when you're changing it in the oven. So what we did was we set the laser up at the top, and we would look through the top of the oven and slide the plate over and get it lined up perfectly. And we had to practice doing this a lot. Ended up standing on a counter trying to get in there and make sure it was exactly right. And the cells can't be out of the incubator for more than 2 hours, so that's why it's so important that we do this quickly. So we kept the oven at 37 degrees Celsius. Again, that's human body temperature, so that's why we're looking for that 37 degrees. We use the 808 nanometer laser, and it's an 800 milliwatt laser. If you'll recall, I mentioned that near infrared is not harmful at low power densities. The standard that is supposed to be healthy is about 1 watt per centimeter squared. Our laser is about 2 watts per centimeter squared. But what we're seeing in the research right now is much, much higher power densities, sometimes even as high as 15 watts per centimeter squared. So we're at a much closer to a healthy range in our experiments. We irradiated each of the cells each of the wells for 20 minutes, the same as we did our photothermal properties. And we did 4 plates with each type of sample. So we're 4 of the thulium NGQDs, 4 of the molybdenum disulfide, and 4 of the GQDs. And the reason we did this is because we wanted to make sure that this was reproducible, and it wasn't just one fluke plate of cells that worked really well or didn't work really well. And then we ran each of those plates in the MTT assay 5 times. So when as many of the cells are dying as are in the really high toxic cells, there's not much absorption, it's very low. And so they need to make sure that the machine is actually reading it correctly. So that's why we ran it 5 times, because it's kind of it's hard when it's that low of a number for the machine. So this gets us into our data. And what we see here is the cell viability in percent on the side, and then we have the DMEM that got irradiated with the laser, followed by the DMEM that was not irradiated with the laser. And that same pattern follows for the water, for the low concentration that originally yields 80% viable cells, and for the high concentration, which is what we used in the photothermal properties. And this is going to be the same setup for the next couple of graphs as well. What we're seeing here with these stars, if it has 1 star, it means that it was statistically significant from DMEM. And if it has 2 stars, it means it was statistically significant from water. And if it's red, it wasn't with the laser, and if it's black, it was with the laser. So you can understand the comparisons that we're making here. So what we're seeing with the thulium NGQDs is that there's no statistical significance between either of the low concentrations and the water or the DMEM. So that tells us that it's not really dying in the low concentration from toxicity, and that it's not dying from any photothermal properties. Otherwise, we would be seeing a difference here. What we also notice with the thulium is that actually our low with no laser has more than 100% viable cells. And so the reason why it has that high number there is because of, as I said before, we're using the control to determine what is the 100% viable cells. So when we see that this has more, that tells us that it did better at surviving than the DMEM with no laser treatment. And what we think is the reason behind that is because glucosamine is the precursor for NGQDs, that it was using that sugar in it to break it down and to feed the cell, so it was actually helping its growth in this low toxic state. Now we do see a significance between the high concentration and the DMEM and the water, and the reason we think that is due to the toxicity of the solution, because we're not seeing anything similar in the low concentration. For molybdenum disulfide, we see the same with the high, that there is significance between the water and the DMEM. We also have to remember how toxic the molybdenum disulfide was originally. With the low concentration and the no laser treatment, we do see a difference between the water and the DMEM, which tells us that this was still a relatively toxic sample. And then in the low, we only see a difference between the DMEM and not the water. So that's telling us that if there was any photothermal effect, that we couldn't totally contribute it to the molybdenum disulfide NGQDs, that part of it is due to the water in the solution. With our GQDs, we get similar statistics as our molybdenum disulfide, but if you'll notice the difference between the low that got treated with the laser and didn't, and the high that got treated with the laser and didn't. We see something very different there, and I'll get into that in just a minute. But just, again, probably this is much more toxic than this one, or we know it's much more toxic, but probably what's going on here, the reason why this is still significant from the water and from the DMEM is the toxicity of it. And this one here, we see that it's significant from the water and the DMEM when it's not treated with a laser, so we know they're dying from toxicity. But then we see with the laser, it's only significant from the DMEM. So if there's photothermal properties that are causing any of the cell death here in the low, part of it is due to the water. So keeping in mind these 4 bars right here on each of the graphs, we look at the statistical significance between the low concentration that was irradiated with the laser and wasn't irradiated with the laser. And then we also look at the significance between the high that was irradiated with the laser and the high that wasn't. So what we're seeing with the thulium is that the low concentrations, neither of them are significant from each other, and the high are not significant from each other. So that's telling us that there's not really anything we're seeing from photothermal properties. Otherwise, the laser concentration would be much worse. For the high, for the molybdenum disulfide, we're also seeing the same thing. It would be much less, and there would be a greater statistical significance if that was the case. And with the GQDs, the high isn't significant from itself, but the low is significant from itself. So the idea that we're kind of getting from this and what we're theorizing is that the low concentration is partially decreasing in cell viability due to the water and to the GQD's effects. And the reason why the high isn't, we're thinking it's because it's just so toxic that we're not being able to see the photothermal effect as much because the cells were going to die either way based on the toxicity of it. So this tells us that GQDs are actually something that we might want to look into in the future. And in the future, we would like to look at doping the GQDs instead of only doping NGQDs. Right now, the lab doesn't have a process for doping GQDs, but that's something they can look into. Or they might functionalize the GQDs with other materials. So like the erbium and the ytterbium, which we didn't use earlier because they were too low with the NGQDs, they might do better with the GQDs. It might be something we would look into again in the future. After that, they would want to look at in vivo experiments, and then we'd like to combine with the research on targeting so that not only are we able to kill the cancer cells, were able to find them and then kill them specifically. The spiritual impact that this had on me was, it was a different way of getting to know God's character. I think when we think about physics and God, a lot of times we think about cool astronomy stuff and how great the universe is and how big it is, and we see this really massive scale and get this idea of how awesome God is. And in this case, I got to look at really small stuff all summer and how those work together and the amount of detail that he put into that. And just there were so many things, one on top of the other, that it was so hard to comprehend, even with people explaining it at me every single day. And so it was just a very interesting perspective, a different perspective on who God is and what kind of attention he pays to detail. Another thing is, a lot of times we think of scientists and think if they're not Christians, they're probably just atheists. And what I found in this community at Texas Christian University was that not very many of the people I worked with were Christians, but I wouldn't say that very many of them were atheists either. There was this kind of idea that a lot of people talked about the universe just working itself out, that there was some kind of this flow or this spiritual idea going on that wasn't really related to a God. So that was interesting to think about, and not having to go in and say, well, first I have to make you believe that we need a God, or there has to be a God for this to work. But being able to go in and say, okay, so you think there's something going on, well, this is who the real God is, and this is why. And being able to have that kind of conversation makes it a different mission field that I wasn't really expecting it to be. And so that's interesting. And I had the opportunity to work closely with a bunch of other REU students and build some relationships with them that I'm hoping will last. And there was one other girl who was Christian there working with us, and so we're just hoping that over the years, it's a small field, we'll all see each other again at conferences, that we're able to just impact them that way. I'd like to acknowledge that this program and this research was supported by a grant from the National Science Foundation to Texas Christian University. And I'd like to thank again and just acknowledge Dr. Nomov, who was my advisor this summer, and Dr. French Boy, who runs the program at TCU. Thank you so much for listening to my presentation, and I'd be happy to hear any questions. Yes.
So thank you, Tatum. I learned a bunch of stuff I didn't know. And that's one of the neat things about being at Laterno. You learn a bunch of stuff you don't know, whether it's knowledge-based or faith-based. So thank you for adding to both in this presentation. I'm curious what, you said future work, but I'm curious what Tatum might want to personally do in this area, either in graduate work or in work after Laterno.
Sure. Yeah, I thought the biophysics was really, really interesting. I had originally thought I was interested in material science, and I got into this and realized I had to read some material science articles and realized that wasn't for me. But the biophysics is very interesting and might be something that I'd pursue further. But I also got to learn a bit about optics, and so that's definitely something I want to look into more as well.
So, this is kind of two questions in one. So you said the TM NGQDs, whatever the acronym was, peaked before the near infrared, but then you said that you used it in the near infrared. I was wondering why I didn't try to use it right, like when it peaked, and also like the other question is like what is the significance of peaks? Because I know one of them just had a kind of steady one throughout. Right. So yeah.
Okay, so let's do the first part first. Why we used thulium and irradiated at 808 instead of 790 where its peak was. Okay, the reason why we did that is, I think I can scroll back to it. Sorry. Okay, so if you'll see here at like about 808, we're still seeing something above 0, and it's absorption. So what's really important is to stay in the near infrared because it's less harmful to people, so that's why we don't want to go back to 790. Near infrared is 800 and on. Also, the only laser we had was an 808 nanometer laser. So that was another reason. And the significance of peaks is just this is where it absorbs the most. So if you're looking for emission like in fluorescence, you want to excite it where it absorbs the most, and that's where, and then it'll emit at a different wavelength. Mm-hmm. Yes.
So if you were able to target the cancer cells and then heat them up, would that heating affect healthy cells around that cancer cell, or does the heat not dissipate that much?
I actually don't know that much about how the thermal stuff works in that. I'm not sure. I would think that you're going to get some heating in the surroundings because they're very close together, but we're not seeing it heat up a ton. So if it's not heating up a ton in that one area, then it probably isn't going to spread to be enough that it's going to hurt the cells, is what I would think. There's a lot of research out there right now where they're trying to get it to heat up like 10 degrees at a time. So that might be something that they might worry about more than we thought about. Mm-hmm. Yes, ma'am.
So Tatum, is is this just the beginning of research, and is it, you had mentioned at the beginning, is it directed primarily to brain tumors?
Mm-hmm. Yeah, so this is just the first thing that this lab has done with photothermal therapy. They have been working with targeting and drug delivery with the graphene quantum dots before. So the photothermal therapy is new for us, and so this is just the first 10 weeks that they've done it all with photothermal therapy, so there's a lot more they want to do. And they are interested in the future getting it to be something that could be used with brain cancer. Yes?
You might have mentioned this, but what made you choose those specific 3 rare earth metals?
So that was when we did the photothermal property test. So first we had to look at the absorption, figured that out, but then once we looked at how much the temperature changed, those were our 3 that had the highest temperature change, so that's why we went for those.
So for as far as concentration of the cells in the tumor and how that affects the area around it, you said the cells die around 42 Celsius, which is approximately 108 degrees Fahrenheit. How does that kill only the cells based in the tumor and not anything else around it?
Right. So I think Matthew was asking kind of a similar question, and you're kind of asking about how it's affecting the cells around it. I'm really not sure. We didn't go into how that affected around it. If you email me, I can ask the people I work with and see if they have any ideas. So,
Okay.
Yeah. Yes?
Graphene quantum dots selectively go to tumor cells, is that known?
No, so that would be what we would do with the targeting. So it's really easy for us to, graphene quantum dots are really good carriers. So I believe what happens is in the cell, or cancer cells overexpress certain proteins. So they're putting receptors onto the graphene quantum dots. And so more graphene quantum dots are going to go to your cancer cells than to the healthy cells that are around it. And so that's what they're working for with the targeting. Yes?
Tatum, you might want to, just for the benefit of students who are interested in applying for REU positions, let them know when you started your search process and how you applied for an REU, and if you've got any recommendations for applying for REUs for students here, that would be nice.
But if it's a competition base, she might not want them to know.
Thanks, Dr. File. Yeah, so I started applying for REUs in December. If you're interested in that, a good resource is the National Science Foundation. Go to their website and look up their REUs. They'll have links to all the schools that are offering REUs. They'll have links to their websites and how to apply to their programs, or you can apply through that website. Just depends on how the specific school's doing it. Also, get in touch with your professors. Dr. Ball actually emailed me the one for TCU because they emailed him, so I wouldn't have applied for it, except it just came through, and I was bored over winter break and was like, sure, I'll apply to something else. And that's how that went down. But you want to get on it kind of early because you need reference letters, and you'll have to write some information about why you're interested in it. So it helps to look up the professors at the university that you're applying to and kind of read about their research, look up some of their papers, get an idea of what they're doing. Because if you can say why you're really interested in it, they're more inclined to want you there. Yes?
Why are there only 2 laser sizes with the 808 and the 980? Has nobody like invented one in the middle, or?
We just don't own more than that.
Okay.
Yeah.
But there is like other
Yeah.
sizes?
So we actually have a 780 nanometer laser. So that's another size that there is. It's just outside of the near infrared, so we didn't look into it. Yeah.
All right, cool.
There's others.
Thank you.
Mhm.
I have a question. You may have touched on this, but the near infrared, as far as brain tumors, how far into the tissue will that go before it's intercepted, or?
I think it can penetrate a couple of centimeters.
Me again. So you said that you weren't able to use tungsten carbide because it wouldn't dissolve in water.
Mm-hmm.
I was wondering if there's anything else in the research that, like, I was wondering why, like, that was the main issue, and if there's anything you are trying to do to try and be able to use tungsten carbide in the future.
So with tungsten carbide, we have a device called a tip sonicator. And what it does is it breaks up your material into really, really tiny pieces. So we took it, put it in water, tried to dissolve it. We put it on a vortex machine first. It just makes it spin really fast, vortex. And then we put it in the tip sonicator, and we would leave it in there for like an hour, and it still wasn't breaking down enough to dissolve in the water. So then we added surfactant to it. We added 2 different kinds. I don't remember them off the top of my head. But we would add those to it, and what that does is it increases the toxicity. So there's only so much we were willing to put in there because we knew it was going to be too toxic for the cells. So eventually, even with something in there to break down it and using the physical method of it as well, we weren't seeing it dissolve well enough to make into the quantum dots. And if it's not dissolved, it's not going to dope onto it. That was another problem with the cerium oxide nanoparticles, is they just would not dissolve in water at all. Yeah.
How were you able to measure cell viability?
So that's with the MTT assay, and what it's doing is when we put something in there, if the cells are alive, it goes through a process that produces the formazine, and then we see how much of the light is absorbed, because formazine absorbs light at 590. So if it's absorbed, then we can determine, based on how much is absorbed comparing to our control, we can determine how much of that cell is viable. Does that make sense?
Any more questions here? That was a really nice set of questions. You guys sound so smart. How did we hear? Smart children, smart students, and smart professors, smart speakers. This is all good stuff. If you want to hear more, Tatum will be here after we close, and we're also going to be having lunch with Tatum in the Joyce Room of the Corner Cafe. The Joyce Room is that looking room with the glass walls. You come into the Corner Cafe. So if you want to join us there to hear more about Tatum's experience this summer, or what else she wants to do with these things, please join us there for lunch. Let's thank Tatum. And we'll see you all here next week, same time, same channel.
God in your speech.
Oh, yeah.
Good job.
Oh, thank you,
great.
sir.